So the last time we had met, last Wednesday, we had finished talking about the cestodes and the trematodes, right? We had talked about the flukes and we had talked about um, the tapeworms, and so now we're going to talk about the nematodes. The nematodes are the only group of organisms in the parasite kingdom, or the parasite, sorry, not kingdom, in the parasite group that are true worms, right? They are really warm. I mean, they're real worms. They're not anything else other than a worm, right? And so it's important for us to understand that. Um, so when we talk about them, they are round, right? And so this is an infestation of an individual who had roundworms, and so you can see that a lot of times when you have these infestations, all you need to do is take an anti-helminthic drug, and it'll paralyze the worms, and out they'll come of your intestinal tract, right? And this isn't just the roundworms like Ascaris lumbricoides, right? This is well, so it is painful, Nelda. It can be painful, but it's pretty rare, right? Now we do see these in the United States, but we see them in other countries, right? Um, I think I have a video I can send you all of an 8-year-old girl who's having an operation to remove the amount of roundworms that are in her intestinal tract because it's causing the blockage of the fecal material, which is going to lead into all kinds of other problems, right? These things can be pretty serious really quickly. In the United States, if we get one of these, usually it's a few of them. It's not very many of them, but sometimes we see them in the feces, and if you're having a little bit of irritation in the intestinal tract, you go to the doc and they do a fecal smear, or they do a fecal occult. Then what they can find out is whether or not there's a little problem, and then they can go and look, right, and might even do an O and P, right? So an O and P is a test you can order, and that's ova and parasite. And so what they're looking for is they're looking for the ova of the Trematoda, Cestoda, Nematoda group, and they're looking for either the cyst of the trophozoite, the parasite of the protozoal groups, right? And so it's pretty easy to do. They collect three three fecal three specimens of feces over a period of time, and then they look for these organisms in them. Usually we don't see the adult because the adults have ways to stay in the intestinal tract unless you paralyze them, and then if you paralyze them, they will get removed from the intestinal tract, right? And so all you need is one treatment of this stuff, and out they will come, right? So this one has been known to not only infect the intestinal tract, but can also infect the lungs and other parts of the body. I had the unique experience when I was working in a clinical setting for a physician. A lady came in and said she had problems with her lungs, and her throat was being very scratchy. And so I took a history, and she said, it feels very weird right now. I look back there, and there was a roundworm in the back of her throat, right? And so I called the doc over, and the doc extracted it. But we treated her with an anti-helminthic drug, and she seemed to recover very quickly. So these organisms can get into lots of different places of the body, right? We believe, our hypothesis was that the lady had consumed fertilized ova of these organisms, and when she did, she aspirated one of the ova into her lungs, and then in her lungs, the fertilized ova developed into an adult and was searching for nutrients or for a mate, and so ended up climbing up through the lungs and up in the back of the throat. That's where I first noticed it, right? So it was a pretty interesting case, but there was no negative outcomes for the patient. We believe she had that one worm, and that was it. And after that, then she seemed to be just fine. Okay. Ascaris lumbricoides, right? It is a roundworm. So you can see the difference here. This is a dioecious organism. Right? It has a male and a female. The female is larger, right? So you can see the female is larger. The male is more slender and smaller, about 30 to 40% smaller than the female. And then you can see this little hook at the end, and that's the indication that it's a male. Does anybody remember why why the females are typically larger? Can anybody remember why? We talked about that before. She's got eggs in her, that's right. And so the technical term is she's gravid. That's correct. Right? So she's gravid. Very good. So the next organism is Nematoda americanus. This is the hookworm. So the hookworm, we have cases of it in the United States, especially in ranching communities more than anything. This is one that maybe your grandparents, or grandmother maybe told you, now, Mercy, don't go running around in the fields barefooted, right? And so the reason that is because the immature form can pierce the tissues in between the toes and get into the body, right? So this particular organism, like a lot of other organisms belonging to the the either the trematode, nematode, or cestode, will cause an increased number of eosinophils to show up, right? That's the white blood cell that shows up um, to fight off the parasite, right? But also you see those in allergies. So there are a lot of people who believe that if they get an infection of roundworms, that they're not going to have problems with allergy. It's become such a big deal that there's a tourist industry now where you can buy a package and go to a country where you can get infected with these roundworms and then bring them back in your body. And then the hypothesis is for those individuals that if they are infected with roundworm, right, either this one, Necator, or another one called Strongyloides, that they're not going to have problems with allergies. So maybe that is true. I don't know for sure. There isn't a lot of real research going on about, which one is better. But to me, I would much rather have allergies and try to manage the allergies with either an antihistamine or a vaccine than to get infected with a roundworm, because they can do a lot of damage. Here is an image of okay, I missed that. James, hold on a second. No, so the I believe the hypothesis is that if you have these high eosinophils, that they block the ability for the allergies to be. Yeah, so I guess what you're saying is correct, James. I guess what you're saying is correct. Yeah. I yeah, I'm with you, Justin. You know, we have antihistamines and other things like that that we can use to manage an allergic season. So yeah, I would agree with you. I do have a friend who is a musician, and he spent quite a bit of money to get infected with these worms. And I haven't talked to him in a while, but I need to follow up with him, because for the first 3 months he swore he didn't have any problems with allergies. So I don't really know. I just think it's kind of interesting that people would think that is a viable way to to get treated for allergies. I just think it's pretty crazy. But you can see here where the hookworm and other organisms in the same group have these things that look like teeth. Now, they're like teeth. They act like teeth, but they're not composed of the same type of material that your teeth and my teeth are composed of, right? But nonetheless, they allow these individuals, you can see where the tooth is here, the teeth are, and where they're attaching to the organism, right? It's right there. And so that allows them to attach to the organism and then take nutrients from the organism. And so that's a pretty serious problem to me, if you have several of these organisms and they're all chomping into your intestines, right? So that could be really, really problematic to me, right? Not everybody thinks the same way I do, yeah, that could be problematic, right? Trigonosporalis is another really dangerous organism, and we do see rare cases in the United States, but it is in other parts of the world that we see these. Trigonosporalis is typically associated with meat that isn't cooked properly. In the United States, it'd be kind of hard to get an infection from like meat that you buy at HEB, because a lot of it is checked through an audit, right? So there are auditing processes that check to make sure that the meat meets certain standards, and they grade the meat, right? So you get grade A, but you can get all the way through grade D, but edible, right? So, but I'm more worried about wild game. So if you are, um, yeah, so I'm more interested in wild game, like for instance, if you a deer or things like that, right? I don't know if you guys ever watched the show Monsters Inside Me. It's fascinating, and for the most part, it's really pretty true. That individual they have there, the beat that's the narrative, is well coached or has a lot of knowledge of parasites and other infectious agents. But they had an episode on there where there was some individuals who were up in Alaska, and they were hunting caribou, and when they had this really great guide from one of the local villages, uh-huh, and what they were doing is they had a camp and they got attacked by a black bear, right? And so the guide shot the black bear. There's a law in Alaska that basically says that if you shoot a bear, you have to report it to the state government, right? And so they reported that they shot the bear. They took the hide and they gave the hide to, I don't know, some group of individuals that take the hide of the bear, that if you kill one, you have to turn them in. But then they had all this meat, and they decided not to let the meat go to waste. So they had a big barbecue, and they invited a whole bunch of the villagers that were around the area to partake in this big cookout, right? And unfortunately, quite a few of those individuals came down with trichinella, right? This is an organism that gets encysted into the muscle tissue, and it causes a skeletal muscle infection, a really serious, painful infection. And this is why pork gets so much of its bad rap, right? Because, hogs, pigs are kind of a cute little animal, but they sure do eat anything and everything, including you. If you're around and they're hungry, they're going to eat you if you're not moving, right? So, this animal, the pig, gets kind of a bad rap because they think that it's the organism that's most associated with trichinella, but that's not the case. The case is that wild game is much more likely to have been infected by trichinella. And then if you eat wild game but don't cook the meat properly, then you can come down with an infection of trichinosis. Okay, questions? All right. So in lab in the last unit in lab, which is a couple weeks away, we will be looking at the morphology of these organisms and how you identify them. So that'd be kind of cool to wrap up the semester with looking at parasites and fungi, I think. Okay. All right. The next organism is Anaerobus vermicularis. Anaerobus vermicularis is the pinworm, and almost everybody has had pinworms, right? It's a very common infestation, and so it usually is in the lower GI tract, right? So the female will come out of the anus when the body is at rest and a little bit cooler, will come out of the human anus, will lay her eggs and then go back into the anus. Sometimes she gets confused and crawls down your leg instead, and sometimes you can feel them and you turn on the light and lo and behold, there is a female Anaerobus, a pinworm, um, on your bed sheets, right? So the way that people sometimes will look for these things, right, is that when the baby's asleep, because usually it's kiddos that get infested by this particular organism. When the baby's asleep, they'll go in there with a flashlight, and what they'll do is pull the underwear down of the baby, infant to child, and they'll spread the cheeks and look to see if the female inner robus is out and laying her eggs, right? And so then they know for sure that's a problem. There are other signs that other symptoms that somebody might be infested with inner robus. Their anus itches a lot, and they're forever trying to you know scratch that itch back there. And so sometimes if you see little kids kind of scratching back there, you might want to get them checked out. And these things are so easily communicable that if one person in the family has it, probably everybody has it also, because they're prolific in laying their fertilized ova, right? And so that's everywhere. So everybody in the family gets treated with an antihelminthic drug. It paralyzes the worms, and out they come, right? Not a big deal. And so it's not a problem usually. Usually the worst thing it does is causes a little bit of anal itch, but in some cases it can cause the prolapsed rectum. This, ladies and gentlemen, is an inflamed rectum, and so you can see it's so inflamed that it has come out of its cavity, right? It usually, the anus itself is an innie, but here you can see it's an outie, and you can see this is a very bad infestation. You can see all the worms all over that anus there, right? Yeah. So almost everybody's had this, and it's very common. So it's not one of those things that we worry too much about, because we can treat it very easily and there's never any real problem. So you can see what the worms look like. This is a female, but you can see how they lay their ova that's fertilized. And so you can see, I don't know if you can see them, it's kind of small, but you can see the ova here, and then you can see the worms kind of in the lower part of the intestines, and then they come out the anus, lay their eggs, and they go back in, right? So, I mean, it's usually not a problem, but people make it a big problem because they will basically say that it's associated with being not very hygienic or whatever, and that's just simply not true. You can pick up pinworms just by being in the wrong place at the right time, right? Because everywhere you touch in public, there's a little bit of feces everywhere. People are not very clean, and so there's a little bit of feces everywhere. And in some cases, there can be these fertilized ova out there, right? Now, I am not a germaphobe. If you see me in public, I am not gonna. I'm not gonna be like Sheldon, who doesn't want to touch anything. I basically will touch just about everything, like doorknobs and handles and things like that. Just not a problem for me. But some people are very, very weirded out by the fact that there's a lot of potential contaminants out there, microbiological contaminants, and therefore just simply try not to touch for anything. Or they're using hand sanitizer all the time, which, as you know, hand sanitizer is really great for viruses, envelope viruses, but not for anything else. But yet people think that, and so they're forever using the hand sanitizer, which is not that great. Okay, complicated life cycle as the general theme for most of these parasites is they have a very complicated life cycle. I did want to make mention of heartworms. Also, a roundworm belongs to the genus Dirofilaria, right? So Dirofilaria, oh heck, I'm just gonna Dirofilaria, right? So this is heartworms. Usually they're picked up by animals, dogs, the ones that we're interested in, from a mosquito, right? So a mosquito will bite a dog, and if the dog isn't being prophylactically treated for heartworms, they could develop heartworms. And so maybe one heartworm is easily managed. You can treat it, and the heartworm will die. But if they have a whole bunch of heartworms in their heart, they're gonna have severe problems, right? Um, my 2 dogs ago, Peppy, he was a little mixture, but we only we only take in dogs that adopt us, right? So the last 2 dogs we had adopted us. Peppy, um, his owner had passed away, and he needed a home to go to, and we went to visit the family, and the dog took to my wife, the little dog, and so they said, "Do you want to take Peppy home?" And Peppy jumped into my wife's lap, and so we took that as a sign. So we took him home, and we cared for him for the rest of his life. So we got him around 7, and he passed away when he was about 16. But he had another 5 years to live because he was a dachshund beagle mix, and so. But what he did was he had heartworms, and we treated him very early on, um, through a holistic way of doing that. And he was heartworm free, but the damage had been done to his heart. So when he was 16, he started to have a lot of problems. He had syncope. And for those of you who have never seen syncope um, ever, right? People just faint, right? Well, dogs, when they have syncope, not only do they faint, but they kind of scream when they faint. It's very dramatic, right? So they get a little bit excited, and then all of a sudden they can't get enough oxygen to the brain, so the brain shuts down for just a moment, and they basically scream when they pass out. And then they lose control of everything, so they defecate on themselves, they urinate on themselves. So it's pretty dramatic. Well, Pepe developed syncope, and the reason he was developing syncope was because of the damage to the heart. He had developed pulmonary emphysema, which means that the chambers of the heart don't completely remove all of the blood from one chamber to the other, and so there's a lack of blood going through the body, which then leads to, if you get a little bit, if you overexert yourself, then you develop syncope and you faint, and so it's pretty interesting. So Pippi died um, because of end-stage renal disease that was brought on by the cardiac or cardio emphysema, pulmonary emphysema, and that was brought on because of the damage to his heart that it's actually done by heartworms. So if you've got a dog, you need to be prophylactically treating them for heartworms on a monthly basis, right? Some people use the stuff that they put on the back that not only treats for heartworms, but also is a deterrent for mosquitoes and other parasites, fleas, and things like that. For larger dogs, that's perfectly fine. But for smaller dogs, I would recommend. I'm not a veterinarian. I'm not giving you veterinary advice. I would recommend the oral one because it's less. It has a less effect on the little dogs. The little dogs are very susceptible to all kinds of neuropathic problems with the stuff that you put on the back, right? Especially not trifexis. Do not use trifexis. Okay. Any questions about varrofilaria? Okay. The last group of parasites that we're going to talk about are the exoparasites. And so the exoparasites are the parasites that are on the outside of the body, right? And so these belong to 2 groups. They belong to the insects, the insecta, they have 6 legs, and the arachnida have 8 legs, right? Now, the arachnida also are the phyla to which the spiders and the scorpions belong to, but those guys are not parasites. The only 2 that in the arachnida that we're worried about as parasites are ticks and mites, right? But really, when we think about those and the insects that are parasites, right, true parasites, are going to be things like fleas and lice. Although we worry more about them not because they're parasites, they're more of a nuisance as a parasite, right? Yeah, they're there, but okay, they're just causing me to be kind of freaked out because they're on my body, right? But really, the real problem in these particular organisms that are parasitic are the fact that they are vectors to a lot of other organisms, right? So if we think about that, the flea is a vector for typhus and a vector for plague. Um, the mite can be a vector for scrub typhus, and the flea is a vector for, I mean, the tick is a vector for uh, Lyme disease. And so uh, lice is also a vector for typhoid. I mean, there's just all kinds of different things that they carry. And so I'm much more worried about that. And most of us in medicine and research and microbiology or entomology, study of insects, are much more interested in these organisms as vectors than we are as parasites, because they're much more of a nuisance than anything as a parasite. The most common of all of these parasites is Pediculus humanus, which is the body louse, right? So this is what they look like under a scanning electron microscope, right? And this is what they look like if you just put them underneath a light microscope and you take a picture of them, right? So you can see that they have 6 legs, right? They are an insect. Um, Almost all insects will fly. The lice do not fly, right? So they don't fly from one individual to another. They do jump, but they don't fly, right? So this is really pretty common. We are There are 2 insects that we have an outbreak of in Austin right now. If you read the medical journals, right, and they are lice, and the other one is bed bug. There's a lot of bed bugs in Austin, Texas, a lot of them, and so but we'll talk about bed bugs in a minute, right? So, these are easy to treat. You just go to the store and you buy an anti-lice medication, and when you do that, then you put it on the head, and it kills the organisms, and then you have to go and pick them out of the hair, right? So a lot of times when I was growing up, I don't know if you guys know what a flat top is, but a flat top is a haircut where it's real short to the skull, except on the top there's a little bit of a burr up there. But almost all the kids in the 80s, 70s, until it changed to get long hair, right? With the glam bands and things like that, right? Had flat tops, and so but you never had lice, right? But with my cousin who had lice, his father just simply solved that by just shaving his head completely, right? That was the easiest way to do it, because not only do you have to get the adult lice from the hair, you have to go in and get the nits, right? The nits are the ova, and if this is hair, right, they will put the ova with a little bit of glue on the kind of the stalk of the hair, and they'll put the egg here like this, and they'll glue it on there, and then the egg will hatch, and you'll get a whole bunch more lice, right? So it is communicable. People get them, but really it's more of a nuisance than anything. People just don't die from lice; they just get embarrassed by them, right? We have a we have a family here in my neighborhood who has a young girl as a daughter, and the daughter invited 8 of her closest friends from school. Um, and this is now about a year ago, but invited 8 of her friends from school. One of the little girls had lice, and so all of the all of the girls had lice, and so that's not a good thing when you have these long, beautiful manes on some of these young ladies, right? And they had to all be treated, all of the lice had to be removed, right? So yeah, it was an amazing thing. I had a niece who wanted a pony, and so her father finally bought her a pony, and the pony was infested with turkey lice, and they're bigger, right? And so the my niece got on the pony, and as soon as she got on there, we could see the lice crawling off of the pony and onto her and getting into her hair that quickly, like within less than a minute, they were on her. And so she, of course, rejected the pony from then on because of the lice. And so that kind of ended her romantic involvement with wanting a pony, right? Because she didn't want to deal with that problem anymore. Yeah. Mercy doesn't like creepy crawly things that crawl on her, right? So that is the body louse or the head louse, right? It's a different type of louse. Okay, now there's a different type of louse that is a sexually transmitted organism, and that's the crab louse. And look at His Majesty himself. This is him, right? So the crab louse will infest the areas of the pubic area, right? And so um, there have been a couple of papers written over the last 10 years talking about that some scientists are worried that the crab louse might become extinct because a lot of people are, you know, manicuring their area in the pubic areas, right? And so because of that, there are some scientists who believe that the crab louse might become extinct. However, there was a paper written, I don't know, I can't remember the author. Yeah. Yeah. Well, because James, everything has a place in the world, right? So if you don't have crab louse, then what is its role in the ecology, right? So if you take it out, then evolution basically tells us something else will fill its niche, its area. So if we don't have crab louse, then what's going to take its place, right? And so is it going to be worse? We don't know. Uh, okay, Presley. But okay, I get it. I get it, y'all. But I mean, so as a scientist, I don't really want organisms to just disappear, right? But that's part of evolution. Evolution basically says that if an animal or an organism cannot adapt to the environment, then it's not going to survive. That's a tenet of evolution. And so, this is a problem. However, there's been a paper written about 8 years ago that hypothesized that that the crab louse would not become extinct. It would just move to a different area. And the area that they hypothesized that the crab louse would move to are the eyebrow and the eyelashes, because the morphology of the hair of that part of the eyebrow and the eyelashes are very similar to the morphology of the hair in the pubic area. So I don't know if you know this, but the hair that grows on the body, except for the eyebrow, the eyelash, and the pubic area, are very cylindrical. They're round, right? And so therefore, when you have a louse, all they need is kind of a little hook to hold on. Are you with me? And I'll show you that in a minute. But the pubic area, yeah, the pubic hair, those are mites, right? But the pubic hair and the eyelashes and the eyebrow, their morphology of the hair is more oval in shape, right? So if you were to draw these things, right, round would be the hair of the body, but more oval in shape would be kind of the morphology of the hair. They don't look very different, I know, but I'm not that great at art. But so the crab louse needs these kind of pinchers to be able to grasp those oval shaped morphology hairs. And so they have these pinchers, right? So if you look at them closer, right, you can see the pinchers where the body louse only has these little hooks, because all they have to do is hold on to a round structure. That's easier to do. So it's an amazing thing. Yeah, all you need is love, right? Yeah. So yeah. So yeah. So that is if you see lice on somebody's eyebrow or eyelashes, then you might not want to engage in kissing with them or something, right? Because then they could get on you also. And so it's an amazing thing to see how these organisms can adapt, right? Because 15 years ago the crab louse is going to become extinct. Then 8 years ago, probably not, because they're adapting well to hanging on to the eyelashes and the eyebrow. So there we go, right? They found a new home. It's pretty interesting to think about. Okay, all right, let's move on. Here are the bed bugs. And so if you go to bed at night, yeah, yeah, if you go to bed at night and you wake up the next morning and you have all these little welts all over your body, that's not one hungry mosquito, right? A mosquito takes a blood meal one time, and then it leaves and it's happy and it goes off to do its regular life cycle stuff. Are you with me? But bed bugs are different. Bed bugs hang out, not on your body. So we consider them a parasite, but they don't really fit the definition of a parasite, in that they don't live on your body all the time. They live there when it is able to be with your body, right, when you're asleep, right? So the bed bugs typically live in the mattress or some other places, and then you can see them where these people are looking for bed bugs, and so they came in and they found in kind of the this the sewn areas of the bed frame of the mattress. You can see that there's a bunch of bed bugs there, right? And so these things leave trails because they defecate and they excrete blood when they're moving around, and so sometimes you can see these blood spots on your bed or on your sheets or whatever, right? But if you wake up in the morning and your body looks like this, I can guarantee you have bed bugs, right? And usually it's not going to be in your house. You're going to be someplace else, and you're going to be itchy when you wake up in the morning. And what's going to happen is, what you're going to find out is you have an infestation of bed bugs, right? And these bed bugs are they're the great stowaways, right? They get in your luggage, they get in your socks, they get in your shoes, and then you bring them home, right? So there was about 20 years ago, there was a big outbreak of bed bugs in the United States, stemming from a very, very famous landmark in New York, where people were going in and buying souvenirs and buying things, and then taking those bed bugs home with them, right? And James, since I think you kind of lived in that area, I don't know if you ever heard of this before, but um, and so that there was a problem there, right? And so the shops around the area had to close down and sanitize for a while. But the only true way to get rid of bed bugs um, is to really turn up the heat to about yeah, so burn it, yeah. Well, you can burn it, but you can't burn your whole house down, right? But the only way you can do it is turn up the heat to about 120 degrees Fahrenheit for about 4 to 6 hours. And what that will do is it'll dehydrate the bed bugs and they will die, right? Unfortunately, 120 degrees Fahrenheit in your house, you can't get that hot unless you put heaters in there and all kinds of stuff, right? And it will melt most of the plastic in your house, right? So, but that's the only way. These insect companies will come in and they will spray your house, I don't know, 8 times to get rid of bed bugs, right? But that's exposing you to chemicals that are toxic to you also, for the most part. So you have to be really, really careful. Bed bugs um, do not know when they are full, right? So they take a blood meal and they just engorge themselves so much that eventually they start to regurgitate blood. And so you sometimes can see this regurgitated blood on the bed sheets, right? There'll be little sprinkles of blood everywhere where they've regurgitated. And then if you just follow the trail, there'll always be a trail where the bed bugs are, right? And you can follow these little trails. And it's mostly dirt and fat and blood that's on the bed bugs' feet, if you will. And you'll find out where their nest is, and so you can get rid of it. But Lucia, you're correct. If I actually helped a student one time who called me up and said, "Prof, can you come and help me? I have a big roach problem, and my landlord can't get rid of them. Can you come help me?" She was a student of mine who had just finished the course and moved to a new place. And so I picked up some diatomaceous earth and I was going to go over there and help her out. And when I walked into her apartment, I said, "Okay, show me these roaches." And so she showed me the roaches. And these things were coming out of the of the wall sockets, right? They were coming out of there. They were in the lamps. They were everywhere. And they were too small to be roaches. So I picked one up and looked at it. And I told my student, I said, "How long have you lived here?" She said, "This is my third day." I said, "You need to leave." I said, "These are not roaches. These are bed bugs. And this apartment is completely infested. They're everywhere." I said, "They're in your toilet. They're um, in your bed." And this was a furnished apartment. And so none of this stuff was hers. The only thing that she had was her clothes. Yeah, yeah. So I said, "Look, I'll write a letter basically telling your landlord that he needs to give you back your deposit and your rent because he's in breach of contract. And hopefully he'll give you back the money." So anyway, the landlord was coming toward her and said, "Okay, I'm going to get this under control." And I said, "Sir, my name is, whoever, and I said, I'm a microbiologist, and I want to tell you that these are not roaches, these are bed bugs, and you're in breach of contract. And I showed him the bed bugs, and I said, um, you need to give my student back her money because she's not going to stay here. Um, and so he went in and he agreed with me. Mostly, I don't think that, because I think he heard one of the neighbors tell the my student that you needed to call like the newspaper or the television people to check it out, right? And he didn't want that to happen, so he gave her back her deposit and her rent because he was in breach of contract and he knew it, and she was gonna pack up her clothes, and I said, "What are you doing?" She's like, "I'm packing up my clothes." I said, "Oh hell no!" I said, "Leave those here. They're infested with bed bugs. Let them be thrown away or burned, and you'll just have to buy new stuff." I said, "It's a good reason to." I said, "Do you have renter's insurance?" She said, "Yes." I said, "It's a good reason to take whatever money the renter's insurance will give you, which ended up being nothing, but and buy new clothes." I said, "Because these clothes are infested, and so just leave." And so she went to stay with some friends and then got a new place. But that's that big bed bug story, right? But as I was leaving, I told her neighbors too. Now there was kind of a group of people who were. I said, "You all need to hold him accountable because if these bed bugs are in this apartment, they're going to migrate to your apartments pretty quickly, right?" And the owner heard me say that, and he said, "I'm on it. I've got a group coming over in an hour who's going to come in and is going to treat the entire building." And I said, "Keep them, hold them accountable because," I said, "you don't want these bed bugs um, to be in your apartment." So that those bed bugs, that apartment was vacant before my student moved in. So they came from somewhere, right? And they must have come from another apartment, right? And so that's the way they spread, right? Yeah, it can be crazy. It can be crazy, yeah. But it's not anything to freak out about. You just got to deal with it from a scientific perspective. It's a problem. You have to solve the problem, right? And so, well, so that was just luck. Maybe it was the big guy up there kind of intervening, because had she not called me, she would have thought she had a roach problem, right? But I want you to be able to identify these different. And it'll be easy, right? You'll memorize what they look like, because I just think as a healthcare professional, you need to understand when you're dealing with bed bugs or you're dealing with lice or crab louse or ticks or fleas. I just think you need to be able to do that. So those will be some questions on the third exam. I'll just show you an image and I'll say, "What is this?" And you'll tell me what they are. Okay. Let's move on to the arachnids. And so these are the ticks, right? So look at the design. This is nature's beautiful design. Look at that reticulated proboscis, right? When I say reticulated, it has all these little burrs. You see those little burrs like this? And so pierces, when it pierces your body, it's really hard to get out. Are you with me? And the only real way to get rid of um, this tick is to come in here with some tweezers and to put your tweezers right there and then pull up on it until it releases, right? Any other way will maybe leave the proboscis um, in your body, or it might cause, if these organisms are carrying things like Lyme disease, it's going to cause the Lyme bacterium to be injected into your body, and then you're going to have a whole other problem, right? Because the ticks are the primary vector for uh, Borrelia burgdorferi Lyme disease. Okay, so here you can see them embedded in this particular organism. Here you can see that's a little nymph right there, but look at the erythema migrans that it's causing, right? The redness around it, right? And that is a problem, right? Now remember, to me, ticks are more of a nuisance than anything, right? So I told you all I have a little place in Bastrop that I am trying to bring back to life. And every time I go out there, because it's in the piney woods, I get ticks on me, but I never really freak out. I just remove the tick and go on with my life, right? Um, Mercy would probably faint. Are you with me? Because she doesn't like things crawling on her. But it's pretty interesting to think about. Okay. So the other group of organisms are the mites, right? And so uh, Okay, I'm sorry, James, I missed it. No, they do. They do. So amblyomma, the Lone Star tick, no. So up in the Northeast, it's Exodes. It's a different species, but here it's amblyomma. Yeah, they carry Lyme. So James, recent studies have said that fleas could also do it, but there've never been a case that I know of anybody saying that they were bitten by fleas and then developed Lyme disease. So that's just a hypothesis that's out there that hasn't been proven yet. But Lyme disease here in Texas is actually transmitted by the Lone Star tick, by amblyomma. It's a tick. Yeah. No, no, it's here too, James. And when we talk about Lyme disease later in the semester, I will I will show you kind of the epidemiology of Lyme and where it started, kind of in the Connecticut area, or where we think it started, because that's where it was first characterized, and then how it spread through a lot of the country now. Yeah, but that's a really good point to make, James. When you're not sure of things like that, hopefully I'll have the answer, but if not, we'll figure it out. I can look it up, or maybe you can look it up. But yeah, so ticks here, amblyomma is known to be a carrier of Lyme disease. As a matter of fact, James, 20 years ago, uh, the physicians in Texas were not very good at diagnosing um, Lyme disease. That they had to all go through special training in Texas to be certified to identify Lyme disease, because it was being misdiagnosed so much, right? So yeah, um, Rosa, I wouldn't worry about them on the soccer field, okay? No, no, they're so Nelda, that's a good question also. So they're different. I'm going to show you the difference, okay? So the ticks are about they're about, maybe, I don't know, maybe maybe at the biggest they get is maybe uh a centimeter. That might be too big. They're maybe 6 millimeters. Are you with me? But the mites are even so small. And one of the mites, like a chigger, is and I'm glad you asked about chiggers, because I've added chiggers to my presentation this semester. But um, things like scabies are mites, right? And so scabies, if you think about them, Sarcoptes scabii, right? These are mites, and this is what they look like right here, right? And so you can see these images of these little guys, and they will actually burrow into the skin, and they will lay their eggs, and then they'll come back out of the skin, and they will eat kind of some of the debris that's on your skin, right? So exfoliated cells and things like that, which their saliva, when they're eating, causes a lot of itching, right? It's a protein that causes a lot of itching, and we itch like crazy. And so that exacerbates the lesions that they cause. And so it can be a whole big mess. Are you with me? So Nelda, I would rather have a case of chiggers, because the chiggers are not really adapted well to our body. They cause us problems, but then they die. But scabies is a different thing, right? Scabies are well adapted to our body, and you almost have to get treated for scabies in order to remove them, right? So scabies, Sarcoptes scabii. The difference is, remember, that the mites and the ticks have 8 legs, and the insects have 6 legs. Now, it gets complicated because in the early stages of development, what we call the nymph stage, or the early stage, the first instar of the mites, they only have 6 legs. It's when they become adult that they have 8 legs, right? But they do have 8 legs. So scabies is a problem, and as you can see, they burrow into the skin and cause a problem. Chiggers are more of a problem for us, right? So we call chiggers red bug, right? And the reason that we call them red bug is because they produce these little welts all over the body when they get on you, and they are feeding off of you, mostly where the pants and the socks and things come in contact with your body, right? This is what it looks like, right? And so you can see it's got 8 legs, right? That's an adult. Here is a nymph who's attached to the body. You can see it only has 6 legs, right? And so, but it has this really impressive stylosome that can inject itself and then attach to you, and then take blood from you. Now the chiggers, Nelda are not very well adapted to us. So they will cause us problems, but then they'll just die. And if you take a shower, you wash them off, right? Scabies a little different. That doesn't work that way. But here you can see the chiggers are problematic, right? And so this in a lot of the United States, chiggers are a problem early spring or early fall. But in Texas, chiggers are a problem year round, right? And they love Bermuda grass, and they love kind of leaf litter, right? So Rosa, if you're on a soccer field that has Bermuda grass, then you want to be careful, right? So yeah, so 50 acres, man, I would love to have 50 acres. That's wonderful. But I mean, I have 1 acre where I live in Austin, and I get chiggers all the time, right? But this is what they look like. Usually, they're not going to be this big a problem on the entire body, right? Usually, you're going to see them. I think I have a picture here. No, I guess I don't. Usually, you're going to see them kind of on the legs, or maybe in the armpits, or around where you have your undies or where your socks are, right? They're going to be a problem there. But you can see the entire life cycle here, right? They get into your skin or other organisms' skin. Yeah. So people have an allergic reaction to them, and they get these huge welts, right? And in very rare cases, Presley, they can develop anaphylaxis to the chigger bites, right? But they produce a whole bunch of eggs, and the larvae go into the environment, fall off of you, go into the environment, and then they hatch, and then they kind of wait around until they attach to animals, um, like us, right? And I've had several dogs, and I always think they're ant bites, but maybe they're chigger bites, but they just bother the dog so much that they really just kind of chew up all of their skin trying to get rid of it, and they lick it so much it becomes a hot spot. And so you have to be real careful with the chigger, and these are problems. James, since I don't know how long you've lived here, but have you had chiggers yet, Mr. James?
No, I have not personally had chiggers, but my
Okay.
brother-in-law actually had a big issue with them in his backyard. He's got a lot of like live oaks in his backyard, so there's a lot of
Yeah,
small
yeah, yeah,
oaks.
yeah, yeah. Right,
But
right, right. So you're gonna get them eventually, James. Just embrace them and say, okay, it's just a Texas thing. But you're just gonna get them eventually. Everybody does. Mercy, have you had chiggers? Mercy might have fainted by now. Yeah.
No.
Okay. No chiggers yet. That's amazing, Mercy. Okay. Yeah. All right. So I want to address the vector problem, right? So to me, uh, you're right, James. If you never go out, you won't get them. That's true. So I want to address the vector, right? So these organisms are vectors, right? That to me, that's more important than a chigger, right? A chigger is a nuisance, right? It causes these red spots on your body, it itches, but they don't really cause any other pathology other than that, right? But some of these organisms, right? I didn't even talk about mosquitoes yet because mosquitoes are not a parasite, but they're vectors. And so we're going to be talking about these vectors, right? So if we talk about the human louse, it can transmit things like typhus. The flea, right, can transmit things like plague and like typhus. The deer fly tularemia. The kissing bug Chagas disease, right? We talked a little bit about that already. So these things are problematic. There are two types of vectors, right? There's a mechanical vector and a biological vector, right? So a mechanical vector would be things like a roach or a housefly, which really don't take a blood meal, but they crawl around on things and get themselves contaminated with things, right? And then they crawl on your food, um, and leave the organism behind, right? So we're mostly talking about a foodborne infectious agent that would cause things like a viral infection, gastritis, or enteritis, or salmonella, something like that, right? So they were out and about, crawling around, the roaches were, and they got contaminated with salmonella from, I don't know, deer poop, and then they came into your house, and when you weren't watching, they got into a box of cereal and crawled into the cereal, and then left the box of cereal, but you ate the cereal, and the cereal had salmonella, and you developed salmonella, and you go to the physician and they say you have salmonella enteritis, right? Which enteritis means enteritis means you have an infection of the small intestines, right? Salmonellosis is the etiology's cause, right? But they say it's idiopathic. Does anybody know what the term idiopathic means? Idiopathic. What does that word mean? Anybody know? Idio Unknown. That's right, Rosa. So when they say you have idiopathic enteritis, you have salmonella, but we don't know how you got it, right? And so a lot of times, a lot of these infectious agents get into our body and they're idiopathic. We don't know how they got there. They just got there, right? So be sure you know the difference between a mechanical vector that crawls around, gets contaminated, and crawls on your food or lands on your food like a housefly, drops off the organism, and then leaves, right? The other one is a biological vector, right? So these are going to be things like mosquitoes. Mosquitoes in Texas is the most important one of them all because they transmit things like in Texas, um, West Nile virus, Zika virus, and chikungunya, right? So lots of different, and then of course dogs, it's also heartworms. So it's important for us to understand what these things are, right? But so you can see, in the world, right, mosquitoes transmit malaria, and that's much more important than West Nile because malaria is a big killer. Where West Nile, I think, last year during the pandemic, um, the, you know, we just weren't really looking for West Nile, except people like me who are nerdy and like to look at these things. I think we had 180 cases around West Nile in Central Texas last year, and only 2 deaths, I think. But the year before that, we had over 400, and we had like 21 deaths, right? So this is a problem. Anytime we lose somebody to an infectious agent that could have been prevented, it is a loss to humanity as far as I'm concerned, right? Because we understand the organisms, we just don't know when they're going to show up, right? So I'm much more interested in a lot of the exoparasites as vectors, and then the mosquito, right? Because the mosquito is such an important vector, and it's not a parasite, right? It doesn't live with us. It bothers us. It takes a blood meal, and then sometimes it'll inject the infectious agent into our body, right? And there are the three different genera of mosquitoes in Texas, right? And those three different genera, I want you to know this, are Culex. Oops. Right? And so I want you to know these, and I want you to know how to identify them. Probably none of you are going to go into research, but I just think that somebody in healthcare, if people say things like, I was bitten by a bunch of mosquitoes, and you say, oh, do you know what the mosquito looks like? And they say it has these little white bands all over their legs. Oh, that's Aedes, right? And so that's a very aggressive mosquito, right? These things are very interesting, right? So if you want to know what they look like, here they are. This was taken out of a magazine that came to me a while back. It's very, very informative, and so I made a copy of it, took an image of it. But you can see Aedes is the very aggressive one. It's the one that'll come at you, and that's the male. The male makes all the noise, but the females are taking a blood meal when you're distracted by the male, right? So they're pretty sneaky because it's the female that needs the blood to encourage to keep on going with the life cycle, right? But the Aedes, you can see, it's got these little white bands on their legs, right? Anopheles is kind of a rotund little mosquito. It's fat, if you will. And then you can see Culex is a little tiny guy, right? But still. And they have different times of day that they like to come out, right? Some of them like to come out in the early morning. Some of them like to come out at dusk, and some of them are kind of like house feeders. They like to be inside the house, right? And so, but if you can get rid of all the standing water around your house, then you're not going to have that big a mosquito problem. I'm never going to have. I'm always going to have that problem because I have a creek in the back of our property, and so there's always going to be a little bit of standing water, and the mosquitoes are going to like that. But yeah, not all of them, Mercy. Only Anopheles can transmit malaria. So the other guys, Aedes and Culex, don't do that. Only Anopheles. Hmm. So if you remember, we talked about the life cycle of malaria before, and if you'll go on there, you can see that I made reference to Anopheles as being the vector for malaria, right? But look, here's some other things that they can transmit in Texas, right? Encephalitis, dengue fever is another big one I didn't talk about, but we'll talk about it when we talk about infections later. Malaria, but not in Texas. That's in the world. Zika for sure. West Nile virus for sure. Yellow fever, but not in Texas, right? Dog heartworm, yes, and chikungunya, yes, right? Which is my favorite of all words, chikungunya, right? I just think it's so cool. You don't need to know the infections. You just need to know. If I ask, if I show you an image and you see the little bands on the legs, that's Aedes. If I show you an image and it's a rotund, it's kind of a big, kind of chunky mosquito, that's Anopheles, and a really kind of minute, little bitty, tiny little guy is Culex. Okay. Anybody have any questions about the parasites? Excuse me. Okay. We are done with parasitology. Let's move on to virology. No, sir. No, 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 ma'am. No mercy. No. No, absolutely not. But you do need to know a chigger is a red bug, and if I show you a red bug with 8 legs, then need to know that's a chigger. Okay, fair enough. Yeah. All right, let's talk about viruses. All right, so very cool biological agents, right? But they're not alive, right? So let's talk about them. So if we look at viruses, they're too small to be seen with microscopes that we even have at ACC, right? You need to have a transmission electron microscope to see them. So for many years, physicians, because back then you were a physician, you weren't really a research scientist, but back then the physicians knew that there was something that was causing infections that were even smaller than bacteria. We could see bacteria, they just couldn't see the viruses, but they knew that there was something spreading among the population. And you tend to make things up when you don't have a way to figure out what's truly going on. So they use the Latin phrase mal aigre, meaning bad air. And so they would say that individuals had infections caused by bad air, mal aigre, right? That led to things like miasma and a bunch of other things, a bunch of other words, right? But it was Louis Pasteur who first proposed that there were infectious particles that were smaller than bacteria, and he really coined the word virus. He called that a virus, right? And so that name stuck. And then as we got to know the viruses quite well, which we don't really know them very well, we know a lot about them, but we still don't know very much about them, right? Then we know a lot about what they do, and really viruses' sole purpose is to infect. That's all they do. And then they replicate, right? That's all they do. They don't do anything else, right? But in the process of infecting and replicating, they cause us to come down with illnesses as a side effect of their biological processes, right? They're too small to be filtered, so we say that they're unfilterable, right? Because they can go right through the smallest filters that we have, which is about a zero point 0.1 micron. They can go right through them, right? They're nanometers in size, right? They're amazing little non-cellular, non-living organisms, right? Every single cell that is alive, every single organism that is alive can be infected by a virus. And the viruses are very host specific, right? So the viruses that infect E. coli are not going to infect us, right? The viruses that infect our liver, hepatitis B, is not going to infect um, a frog, right? And so these things are very, very host specific, which brings up the idea that I was going to talk about, but Abdullah already sent me a couple of things about this. Is why don't we use bacteriophages, bacteria that will infect only viruses that will infect only bacteria, as a way to treat people who have infections. And that's a really good hypothesis, right? We're just not willing to accept it in the United States because it's just weird that we put an infectious agent in somebody and then allow it to kill off the infection that they currently have, right? People don't want to get it, right? They just don't want to deal with that. But it's being used in other countries, right? And there's some clinical trials going on. And in some cases, when there was no other there was no other way, no other path to help somebody survive, they have used what we call phage therapy, because these infectious agents, these viruses, are so host specific that a virus that infects Staphylococcus aureus is not going to infect E. coli, right? So once they get done with killing off the bacteria that's causing the infection, they just simply would be destroyed by our immune system because they do not have any protein that recognize them as being part of our body. It's a great hypothesis, right? I hope we embrace it at some point, but it doesn't look like we're not going to do it anytime soon because it freaks people out too much. Mercy, you'd be freaked out by getting a virus to fight off tuberculosis, wouldn't you? Yeah. So yeah, Mercy's kind of squeamish, right? Okay. Virus infects a cell. It injects its nucleic acid, either DNA or RNA, and that nucleic acid takes over the biochemistry of a cell, and it redirects the cell to produce new viral particles, new viruses, right? And so a bunch of new viruses are made. So that oh, Mercy, you've had this already in 1406 or something like it, right? But so you get a whole bunch of viruses that are made, and the cell breaks apart, and all the new viruses get released, and they go on to infect other cells. That's why you can you can be exposed to flu on Monday and then have symptomology by Thursday or Friday, right? Because they move surprisingly fast. There's a lot of different morphologies, and you're saying, Provi, how did these people ever come up with these drawings of them? Well, these are all drawings for sure, right? And they're quite beautiful, right? To me, they're almost pieces of art, right? Just look at them, look how beautiful they are, right? But these images were inspired by micrographs. So these are all micrographs. These are all electron micrographs, transmission electron micrographs. And then you can see how we figured out what the morphology of the viruses are, right? So if we think about them, right, there's so many different properties of viruses. I'm only going to ask you to know 3 of them, right? So the 3 go in this order, right? Number 1 is nucleic acid, right? That's important. So number 1 is nucleic acid. Is it so? It's either uh, it's either DNA or an RNA virus. So which nucleic acid? Secondly, it's enveloped or non-enveloped. Sorry, my dyslexia is giving me troubles today. And then 3, it's morphology. Now there's a bunch of other things, right? So what type of enzymes does it have? It could go on and on and on. But these are the only 3 for this course that I'm going to hold you to. So in order of importance, right? It's nucleic acid. Is it a DNA virus or an RNA virus? Okay. Yeah. Um, those are 3. Yeah. So second, is it enveloped or non-enveloped? Envelope means it has a phospholipid covering. Non-envelope means it just has a protein covering, right? And then the morphology, right? What does its shape look like, right? And so you can see that there's so many different properties, so many different characteristics of the viruses. I'm just going to hold you to those 3, okay? So here, if we compare E. coli, you can see E. coli, if we measure it in length, it's about 2 micrometers, right? Streptococcus is about 1 micrometer. And the smallest bacteria are Rickettsia, and they're about 0.3 micrometers in size, right? But look at the Mimi virus is one of the bigger ones, right? It's 450 nanometers in size, a little bit bigger than, if you will, some of the bacteria, but this one's not a problem, right? You start to get problems when you get smaller, right? So you can see here's herpes at 150 nanometers, followed by rabies at 125 nanometers, And there's our friend HIV at 110 nanometers, right? Influenza is at 100 nanometers in size. And then those are all enveloped, right? And then the rest of them are non-enveloped, and you start to see the infectious agents, adenovirus that can cause things like common cold or cancer, all the way up through, yellow fever that's only 22 nanometers in size, right? Small. A hemoglobin molecule is 15 nanometers in size. So, I mean, just think about the size of proteins, and these things are a complex protein, right? They're about the size of a complex protein, right? To me, that's just an amazing thing to think about, how we look at these things in size, right? They're so small and they don't stain very well that we have to use special staining techniques. We either have to use a combination of stain and metal, or we can just sputter coat. We can just cover the entire tissue with a type of metal, either colloidal gold or colloidal silver. All those processes will render anything you come in contact with dead, not able to infect, but then we can see them, right? And so they're an amazing thing to look at. This is influenza virus right here, and this, ladies and gentlemen, is Ebola, right? Yeah, those are true micrographs, right? So if we think about them, then the virus, all viruses have two parts, right? They have a nucleic acid, and then an outer covering called a capsid. The capsid is composed of protein only. Animal viruses, some of them, not all of them, animal viruses will have an additional layer. Not only do they have the nucleic acid and the capsid, but they also have an outer covering called the envelope. And the envelope is composed of a phospholipids, right? And they steal that from the cells they're infecting, right? As they are leaving the cell, they will steal the they will steal the phospholipid bilayer from the cells that they're infecting, and they take a little bit away from it, and off they go to infect other cells, right? Therefore, when we say that COVID-19 is susceptible to hand sanitizer, the reason it is because it is mostly phospholipid that's protecting it, and the phospholipids are very sensitive to alcohol. And because these things are nanometers in size, right? You just need a little bit to disrupt them, right? Now, the phospholipid bilayer, really, its function is to help the virus get into our cells because it can fuse with the phospholipid bilayer, right? So, and in some cases, if it, like coronavirus, for instance, right? When it leaves the lung tissue or whatever it's infecting, it takes a little bit of the phospholipid bilayer with it, including some of the epitopes, some of the markers that say, I'm part of this body, right? And so now the immune system says, no, you look funny, but I'm not going to destroy you because you have these markers, right? Therefore, we give a vaccine that has other proteins that are part of it, the spike protein, right? And the spike protein really basically say, this is what I'm going to affect. I'm going to attach to the specific receptor site with the spike protein. Well, our vaccines are against that spike protein. So now the immune system gets trained on how to identify COVID-19 and destroy it, right? And you've seen the numbers, right? The vaccines work remarkably well. And so it's really cut down on a lot of the disease and a lot of death. We're still having a lot of death associated with COVID-19, but it's mostly for the unvaccinated individuals, right? So here is McGraw, I'll give them credit. And they have a really nice image showing, the pink is showing the nucleic acid, right, in each of these viruses. Then you can see the morphology is it's got 20 different sides, right? So it's a icosahedral, right? And so you can see all the different sides to it, right? This is the capsid, right? It's the protein covering. Here's the capsid, the protein covering. But this one, we've added an additional layer so that you can tell the difference, right? It's got an envelope, right? So again, remember, the function of the envelope is to help with infection, help for the cells to get in, for the virus to get into cells, but the capsid is really for protection of the nucleic acid. Now, for those viruses that don't have an envelope, the capsid is kind of the way they get in. They have points and they have spikes and they have piercing all kinds of different mechanisms by which they can get into the cell that they're infecting. Okay. So here's an example of the capsid, right? The capsid is right here, right here, and the envelope is surrounding it. All of this is the envelope. Okay, an amazing thing. All right, so you can see these particular these particular viruses down here do not have an envelope. Okay. All right. So here you can see the differences in kind of some of the morphology and the nucleic acid and the envelope all together with these infectious agents here. And then here are the naked ones. We call them naked, non-enveloped, right? Because they don't have a phospholipid envelope. They have either spikes or they have points to which they can pierce the cell. That way, all of them have enzymes that will trick the cell to let them in, right? And so it's like the virus gets to the cell and they say abracadabra, right? And in they come, but it's really their enzyme systems that trick the cells to get into the cell itself. Pretty amazing the way these things work. Okay, it's just a little bit of computer generated imagery for them itself, right? So, in summary, just I want you to know that yes, the capsid in some cases helps the virus infect if they don't have an envelope, but mostly the capsid is for protection of the nucleic acid, and the envelope really is really to increase the infectability of the virus to the cell. Animal cells, right? Plant cells don't have them because plant cells have cell walls, and so they're not going to be really as affected or would be as affected by an enveloped virus. Animals, you and I, will be different than that. We're going to be very susceptible to enveloped viruses, right? So when we look at the characteristics, right, as Rosa pointed out, right, we look at the characteristics, right? We look at the nucleic acid, right? Is it a DNA or RNA virus? And then we look to see, is it enveloped or not enveloped? And then we look at the morphology, right? Some of them are long, some of them are round, some of them are whatever, right? So if we look at the medically relevant DNA viruses, right, they're not that many, right? So if we look at this, you can see here the pox viridae, right, like smallpox, chickenpox, things like that. Then you see our buddies, the herpes viridae, herpes simplex 1, herpes simplex 2, right? Those are enveloped, right? Those are DNA enveloped viruses. Non-enveloped would be things like the adenoviruses, right? The papoviruses that can cause things like human papillomavirus, right? And then you can see the paraviruses, right? So these are, how many? 5 different groups that are DNA viruses that are medically relevant. It doesn't hold a candle to those organisms that are RNA viruses that are dangerous to us, right? So look at them, they get very complicated very quickly, right? So you have RNA viruses, you have enveloped, you have non-enveloped, right? If we look at just and you're not going to be responsible to know these things, right? I'm just showing you how we break them apart, right? If you look at these things, right, you have envelope viruses that are single-stranded genomically, right? And so you have the orthomyxviridae, the bunnyviridae, the arenoviridae here. Then those non-segmented things like the paramyxviridae, the rhabdoviridae, which are rabies, the filioviruses, which are things like Ebola, and then you have the coronaviruses like COVID-19, SARS-CoV2, right? MERS, and all these other infectious agents. The sneaky little group is right here. These are the retroviruses, and there are a few infectious agents, a few viruses in that group that we are worried about. One of them is a human lymphoma virus, and the other one, of course, is the human immunodeficiency virus, HIV. And then the last one is an infectious agent, a cat's feline leukemia virus. But we're worried about it because it has such similarities to HIV that we're worried that someday the feline leukemia virus could become a problem for humans also. All it needs is probably a couple of mutations to do that, right? So you can see just how interesting these infectious agents are, right? And we're going to talk about a few of them, right? So um, in 2000, right, um, I was a very young microbiologist, and I went to a nerd conference, the American Society for Microbiology. I had a couple of colleagues that were big time virologists. I'm considered a bacteriologist, mycologist more than anything, and so I wanted to go to this particular symposia because the virologists were going to be talking about how they were going to come to a standard of how things were going to be named, right? And so I got in with my friend. You had to be a virologist to get in there. Well, I snuck in with my friend, and then they had this big old debate about things. They decided, right? They decided that we look at the chemical compositions, genetic makeup, right, and the structure, right? So the things that I talked about, right? Similarities in genetic makeup, DNA, RNA, chemical composition, envelope, non-enveloped, and structure morphology, right? And they said we're going to have 3 orders, right? We're going to have 63 families, all ending in viridae, and then we're going to have 263 genera, all ending in viruses. So chickenpox virus is a genus. Are you with me? So they all agreed, and then they went back to their research areas or their clinical labs or their medical institutions, and they did whatever the hell they wanted because they didn't really want to listen to anybody, right? So I have a really close friend who's now retired who found a novel virus, and this novel virus is an environmental virus that doesn't do much to humans, but it's a marker for all kinds of other problems in the environment. And he named it the Sasha virus. And the reason he named it the Sasha virus is he named it after his daughter because he loved his daughter so much. And so he gave her that honor of naming his virus that he characterized and found after her. She was delighted, right? Yeah. She was delighted, James, to tell you the truth, because she grew up in a home with a bunch of scientists, right? So she was delighted that he would name the virus after her, the Sasha virus. Look it up; it's kind of fun, right? Pretty amazing, right? So here are the things that they propose, and then they didn't really stand with it, right? But I just look at these, right? You're not going to be responsible for all these. Yeah. So just look at the DNA virus: that's smallpox, cowpox, fever blisters, cold sores, genital herpes, chickenpox, shingles, cytomegalovirus that causes blindness in people, um, adenoviruses infection, several types of warts, right, including human papillomavirus, and then you see erythema infectiosum, right? Those are the DNA viruses. Look at the list of the RNA viruses that cause problems in people, right? Polio, hand, foot, and mouth disease, all kinds of diarrheal infections, all of the encephalitis viruses, Eastern equine, Western equine, yellow fever, St. Louis encephalitis. Then you get into the bad boys, right? Rubella, German measles, dengue fever, West Nile, California encephalitis, right? Then you get into the really bad boys, right? Rift Valley fever, Crimean Congo hemorrhagic fever, like Ebola, right? Ebola, Colorado tick, rotaviruses, right? Parainfluenza, mumps, measles, the common cold, right? Which it's really interesting because when HIV first became a big thing, the Reagan administration said that they were spending millions on studying HIV, but what they were doing was they were studying the common cold and saying that that was related because common cold causes immunosuppression, right? So if you got a common cold, you're going to be immunosuppressed and you can come down with secondary infections. Almost all viruses cause immunosuppression. It's pretty amazing, right? So then you go on, you see influenza, mumps, measles, uh, there's AIDS, right? Lassa fever, and then you see our corona virus, enteritis, and severe acute respiratory syndrome, which is SARS, which is the COVID-19, right? And so these are an amazing list of killers. Viruses are the biggest killers in the entire world, right? They are not the biggest killer individually. That's malaria, right? Malaria kills more people on an annual basis than a lot of other things, except for one infectious agent that nobody's scared of, and that's influenza. Right? People are not afraid of influenza, but influenza kills a lot of people on an annual basis. It's an amazing infectious agent, yet people are not scared of it, right? And they don't get vaccines against it, right? I get vaccines every year for influenza, and everybody should, but people won't, right? For lots of reasons. So here you can see is an animal virus being infected by this particular virus. It comes in, it uncoats, right? The nucleic acids, the nu- let me go to this way. The virus comes in and attaches to specific receptor sites. This is an animal virus. It fuses with the plasma membrane. It uncoats. Its RNA or nucleic acid gets in and it redirects the cell to make a bunch of viral particles, right? The viral particles come together. It produces a virus. The virus leaves the cell, and when it takes a cell, it takes a piece of the plasma membrane with it, and that plasma membrane becomes its envelope. Isn't that an amazing piece of biochemistry? It's just beautiful, right? Questions? All right. So let's look at the bacteriophage. A bacteriophage is what Abdullah is really interested in because the bacteriophage could be used to really treat bacterial infections. A bacteriophage is a phage, is something that consumes bacteria. So this is a consumer of bacteria. Are you with me? So this is a virus that destroys bacteria, right? So you can see its different morphology pieces, right? It has got a capsid to protect the nucleic acid. Inside of it, it's got DNA. It's got a little neck here. It's got a sheath, and the sheath acts like a syringe, right? It's got these tail fibers, and the tail fibers will actually allow that virus to attach to specific receptor sites on the surface of the bacteria it's going to infect, right? And then it's got an end plate, and it's got these little tail pins in it, and the tail pins will help to really kind of break down the cell wall of the bacteria so that it can inject its nucleic acid into the cell wall. Okay, we're going to use this particular organism as a model to study the infectious process of a cell by a virus, right? And it's a little bit different with animal viruses, right? But it's real easy to understand if I show you this, and you can just kind of say, oh, well, instead of that, the animal virus would have fused with the plasma membrane and would have got in that way, right? Because there's so many different mechanisms to really think about that you just really take the most simple one. And the simple one is really using a bacteriophage as a model, right? And so there are going to be 6 different stages of the infectious process, what we call the lytic cycle, right? But it's the lytic cycle because when the virus infects a cell, it's going to cause the lysis of the cell, and the cell is going to break apart, and then a bunch of viruses are going to be released, right? So the first one is attachment. That's the first phase, if you will, attachment, right? Then there's penetration. Then there is biosynthesis of viral pieces. There's assembly of viruses. So all the pieces come together and they form a new virus. There's maturation of the virus. So when this happens, there's a little bit of enzymes that get released from the viruses. It weakens the cells, or it allows for the envelope to be formed around the virus, and then the virus gets the last phase or last step is release. Oops. Okay. So those are the different Those are the different steps or stages in what we call the lytic cycle, right? So remember, viruses are host specific, right? Now, almost all viruses are going to infect one type of cell, right? There are some viruses like rabies that will infect quite a few different organisms, but they always infect the same type of cell, right? It's going to be the neuron, so it's going to be the nerve tissue. Okay, so in attachment, what you have is a virus, no matter which one. We're looking at a bacteriophage, not drawn to scale, but the virus is attached to specific receptor sites on the surface of the cell. It recognizes those and says, "Oh, this is what I can infect," and it infects and it attaches to that particular cell, right? Then, whatever mechanism, whether it fuses with the plasma membrane, in this case, it drills into the cell. That sheath part acts like a syringe, and it injects the viral nucleic acid into the cell, right? The viral nucleic acid becomes transposonic. What does that mean, Mr. James? It becomes transposonic. What does that mean?
It it makes the bacteria print new viruses.
That is true, but when it becomes transposonic, means it becomes part of the genome, right? Jumping genes.
Okay.
That's right, Mercy. It becomes part of the genome. Okay, and it redirects the cell to not do anything for the cell, right? Oh, you're going to make ATP? Okay, good. But that ATP is not going to be used to grow. That ATP is going to be used to make more viruses. Are you with me? So the nucleic acid gets in, it becomes transposonic. It says, you are now under my control, right? For those of you who watched Star Trek, it's like the Borg. Are you with me? So it takes over. To resist is futile, if you will. Okay, so under the direction of the viral nucleic acid, a bunch of new viral components are made, right? Under that viral component, that biosynthesis, they're all made, right? Then the enzyme systems are there also, right? They have, it has these genes to code for enzymes to put these things together, and so these viruses start to get put together, right? They get assembled, right? By enzymes, right? The enzymes, right? It's all being driven by this nucleic acid, right? Taken over, and the cell is now making a whole bunch of new viruses, right? All these parts come together, right? You have these beautiful new infectious agents come together, right? The new viruses, they've matured. They all release a little bit of an enzyme called lysozyme. Lysozyme makes the cell wall very fragile, right? So it starts to break down the cell wall in different places. And eventually what happens is the cell breaks apart, it bursts, and all of the new viruses get released, right? And it's showing 4, but there can be thousands of these things in there, right? Maybe millions. And so they all get released and they can go on to infect a new cell. And that's why we feel okay on day 1 of exposure to influenza, but day 3 or day 4, we're in deep trouble. Are you with us? So it's an amazing piece of biochemistry, right? And so here's that entire process, right? Some people will refer to attachment as adsorption, right? But you can see how you have absorption, penetration. So this is 1, this is 2. You have biosynthesis, you have assembly. Oops, this is 3. You have assembly, you have the maturation process, and eventually you have release. Now this is showing there's a hole in the cell that it just breaks apart completely, and then the viruses are released, right? Here's an electron micrograph showing a bunch of bacteriophages attached to the cell. Here's an artist kind of illustration that it was inspired by that micrograph, right? You can see how it's attaching to the cell, right? And this is one cell that's being infected or being attached to by a bunch of viruses. And the really cool thing about that is that once one of those viruses penetrates the cell, there's a molecule that's released that basically says, I'm in, and the rest of the viruses release so they don't get wasted. Isn't that interesting? Beautiful. We can actually quantify the amount of viruses by doing a plaque assay, right? So we've made a lawn on this particular plate. We've made a lawn of bacteria that the virus is susceptible, or that's susceptible to the virus. And everywhere you see a little clearing around on that plate, that is where there's an infection of a virus. So you count all those plaques, all those clearings, and you have the number of viruses, and then you can multiply it by the dilution factor, right? So we do this exercise in the majors microbiology course, but we won't talk about it in this course, right? It's a little bit beyond the scope of the course. Okay, questions? So here's a bunch of images of a bacteriophage so that you can see that these aren't just things we made up, right? The shape of that bacteriophage was inspired by these transmission electron micrographs, right? And so we actually used, I didn't, but the scientists that were, they actually used a type of dye that would be very attracted to the nucleic acid. And so you can see in that capsid, you can see that nucleic acid lit up, right? So it's pretty interesting when we think about how we can visualize these things, right? So there is a lytic cycle. And what's the purpose of the lytic cycle, Miss Mercy? What's the purpose of the lytic cycle? Anybody can answer. What's the purpose of the lytic cycle? To replicate viruses. Very good. The other part of some of the cycles, that's right, James, replication of the virus. The other part is called lysogeny, or the lysogenic. So in some cases, right, we already said that the nucleic acid becomes transposonic, right? And Mercy said that means it jumps into the chromosome. So here you can see that. But then what happens with lysogeny is that the viral nucleic acid just goes dormant. It goes dormant, and every cell that is propagated from that particular parent cell thereafter, and all the cells that come from it, all the progeny, will have that particular that viral nucleic acid in you. So for those of you all who do not understand this, that means that every time you get infected by a virus, that a little bit of the viral nucleic acid stays with you. So it's an amazing and beautiful piece of biochemistry that this happens. This is really important for things like chickenpox and herpes, right? So both of those, there's a lot of them, but both those are the most common, right? Both of those they'll cause you to be infected, and then they go dormant, right? And then for some reason, you get stressed out later in life, right? And you can develop with chickenpox shingles, or you can develop an eruption of herpes, right? So you can have an eruption of herpes either urogenitally, which we basically say that is, herpes simplex 2 or herpes simplex B, and then herpes simplex A is when you have a cold sore, right? So around the mouth, usually. And so you can see these things when people are really stressed out. Stress brings on the turning on of the lytic cycle, right? So this lysogenic, right? And every cell that gets divided from there, every cell that's produced after it's become lysogenic has that viral nucleic acid. But at some point you get stressed out for whatever reason. Could be that you have a lot of tests. Could be that Provi is driving you crazy. It could be you're going to have a baby. It could be you're going to get married. It could be you just got a lot of sun one day. Are you with me? Those are all stressors to the body. And so therefore, the lysogenic prophage, that nucleic acid that has become transposonic, turns on, and it goes into the lytic cycle, and the entire lytic cycle process happens, and then you have a bunch of viruses being produced. And in our case, with chickenpox and herpes, we have an eruption of either shingles, right, with chickenpox, or we have an eruption of herpes, either simplex 1 cold sore or simplex 2 lesions around the urogenital area, right? It's interesting to note, but not surprising, that a lot of times when you have when an older person has shingles, the shingles virus is going to be appreciated on the body where the nerves are. Are you with me? So it's going to typically follow kind of one of the nerve pathways of the body. So sometimes you can see this on the chest. Sometimes you can see it around certain parts of the head or the face going into the scalp. Sometimes you can see a long streak. It's a streak down the back, but it's not going to be diffuse. It's not going to be like it's covering the entire body, right? Because it's going to be associated with where those nerves are, right? And so that's a really important thing to understand. Be sure you know the difference between the lytic cycle and the lysogenic cycle. Okay, let's go on. Cytopathic effect. So cytopathic effect is what happens when a virus infects a cell. It's going to affect the cell in different ways, and there are all different types of changes that happen, right? The cell can have inclusion bodies, they can round up, they can act different, right? So for instance, ladies, when you have a pap smear and they come back and say that they've noted dysplasia or dysplastic cells, what does that mean? When you have a pap smear, what does it mean when they say you have dysplasia or dysplastic cells? What does that mean, ladies? Gentlemen, if you know, tell me. It's abnormal. That's right. So what has happened is you've been infected by the human papillomavirus, and the human papillomavirus has caused the cells to look different than what they normally do, right? And so what they normally will do is they might say, can you come back in, I don't know, 3 months or whatever, and they're going to do another one. And then if it's clear, they're going to say, okay, it's fine. It's been cleared by the body. If you continue to have it, they're going to look at you more frequently because human papillomavirus, of course, can be the leading etiology to cervical cancer in women. And so we don't want that to happen, right? Therefore, again, you might know this already, but I'm a big proponent for vaccines, and I think everybody should be vaccinated against human papillomavirus, right? Everybody, young kiddos like 12 should be vaccinated against human papillomavirus. If we can save one person from not developing cervical cancer, mouth cancer, anal cancer because of human papillomavirus, the vaccine did its job, right? And so it's really important. I don't need you to know all the CPE, but I do want you to know what cytopathic effect is. I want you to know that cytopathic effect are associated with viruses, and they change the cells that they're infecting, right? They cause all kinds of changes, right? I don't need you to know the specific changes. I just need you to know what CPE is. Okay? Questions? All right, let's go on. So I'm going to talk about a few pathogens. I don't think I'm going to finish today. We'll go till another 20 minutes or so, and then we'll stop, and then we'll just finish it on Monday. Your third exam is scheduled for next Wednesday, right? So let's be sure we're all on board for that, right? Influenza, I put this on as number one because it is the most important virus out there, and people don't take it seriously. It's a big time killer, right? The last big outbreak of influenza that killed a lot of people was, of course, Spanish flu of 1918. And Spanish flu of 1918 killed millions of people, millions. I think it wiped out a third of the entire population of the world back then. It was an amazing infectious agent, and it was so easily spread, right? Yes, COVID-19 is an important pathogen, but COVID-19 does not hold a candle to the Spanish flu of 1918. COVID-19 is a warning because we're going to have another major outbreak in the next 10 years or so. And that one's going to be very, very dangerous. It's going to be like this one. And that one's already here. It just hasn't mutated enough. And I'm going to talk about it in just a little bit, right? But you can see influenza refers to flu, right? It's a RNA envelope virus, right? Belonging to Orthomyxviridae, right? And it not only affects humans, but other animals also, right? Influenza gets its name from the Italian word influenza. So it means it has influence, right? But it really comes from Latin, right? The most common symptoms are chills, fever, sore throat, muscle pain, severe headache, coughing, weakness, and general discomfort. The last time my wife and I both got influenza, we both had the vaccine, and I was probably kind of sick for about 2 days, and then I felt better. But she was out for a couple of weeks, right? So everybody's physiology, everybody's immunology is a little bit different, and so the infectious agents are going to infect and affect people differently, right? Um, some people are naturally resistant to influenza and will never get influenza no matter what, right? Just like some people are naturally resistant to SARS-CoV2, COVID-19, and never going to get COVID-19 no matter what, right? But that's really pretty rare, right? And so if we think about this, this was a triage center during the Spanish flu of 1918. And you can see how they put all these people who were sick in one room, mainly to sequester them from the rest of the population. But a lot of people died. It was a terrible time. So when my brother-in-law passed away, we were in a little kind of private cemetery for that particular area where there was a lot of German people who immigrated to that area up in Bell County. And in one section of the cemetery were about 9 people, most of them kiddos, who died from Spanish flu. There were probably more, but I was really kind of taken aback by how young these kids are, right? 7, 6, 8, and that's because that particular influenza virus was indiscriminate. It just killed everybody it came in contact with, right? Serious pathogen, right? The two that are circulating right now is swine flu. You may have heard of it, H1N1. It caused a pandemic in 2009, right? Most people don't remember it because it really wasn't that dangerous an infectious agent. We lost some people, but the people we lost were already dealing with some other comorbidity. They had diabetes, they had cardiovascular heart disease, they had cerebral vascular disease, they had all kinds of problems, right? So it killed people, right? And that's what nature does. Unfortunately, it takes the weak people out of the population, right? So this is what viruses do, right? So it was called swine flu because there was a mixing of antigens. There was a mixing of proteins from humans, one, from birds, another one, and two, from pigs, right? And so, yeah, well, I'm just showing you how these things might happen. I mean, my dog when she was alive was part of the family, and she would come and sit on our laps and give us kisses on our lips and things like that, and we thought nothing of it. But really, there was all kinds of microorganisms that are being shared by her to us, right? Because we don't know where her mouth was, but that's just the way it is, right? But people get crazy about this stuff. They think that you can get swine flu from eating pork or things like that. That's not true. All influenza, all COVID-19, all coronaviruses are spread by droplet nuclei, right? They're dropped by the droplets coming out of the nasal passages in the mouth, right? So when you sneeze or cough or talk even, then these things come out and then they can get you. So this one was a pandemic. It didn't last very long, um, and we didn't even have a vaccine for it. Now we do, right? The vaccine is a lot of times put into the annual influenza vaccine that you can get. You get the three most common. You get the three most common viruses that are causing influenza that particular year in the southern hemisphere, and then they throw in H1N1 because if it shows up again, they want to be ready. Now, they're probably going to throw in COVID. They're probably going to throw in COVID-19 for the next foreseeable future, right? To me, it makes a lot of sense. I think we need an annual booster for COVID-19. That hasn't been said yet, but I'm telling you, we need an annual booster, right? Because that infectious agent is quite dangerous. And there's too many people who don't really believe science and aren't going to take the vaccine, right? So in that case, we just need to have a booster every year just to wake up the immune system. And if the virus has changed enough, we need to have those new proteins in there, and we know how to make them now, right? And that particular piece of the research that was found to be able to make the vaccine comes out of UT, right? And so there's a scientist there who figured out the spike protein and how to make antibodies against it, and he's probably going to win the Nobel Prize at some point, right? Because his work saved a lot of people's lives. It's an amazing feat of molecular science. Okay. The other bad boy of the group is H5N1, avian flu, and it's here already, right? It is so bad, it has a 60% mortality rate, where H1N1 only has a 0.1% mortality rate, right? Didn't kill very many people. This one kills people, right? But it hasn't developed or hasn't mutated in a way that it's easily communicated, right? And so when it does that, we're gonna have a problem, right? Because this thing kills a lot of people. Of all the people who come down with H5N1 avian flu, 60% of them die, right? So that's that. So we think of think of COVID-19, right? It has anywhere from a 3, depending on where you live, to about a 12% mortality rate, right? And well, I'm not trying to be reassuring. I'm just trying to tell you the truth, right? So I'm hopefully I'm not here to make you feel better. I'm here to tell you the truth, right? So you can look this up. It's published, right? These things are published, right? So when we think about that, just think of the United States, right? There are about 325 million Americans in the United States, right? So let's take 1 million of them and say they come down with H5N1, right? Avian flu. How many of them are going to die? How many of those million going to die? 600,000. Now multiply that by 325, and you get to see kind of the problems that we're going to be facing. Besides an epidemic of the virus, there'll be an epidemic of fear. People will be hurting each other for a bottle of water, right? Yeah, it's been like that throughout history. History will repeat itself again and again and again because we, the human population, are not smart enough to learn from the past, right? We are not ready for a pandemic of this nature. We were not ready for SARS-CoV2, COVID-19, and look what it did. And even then, James, there are still people, yeah, there are still people who won't get vaccinated. I don't get it, or maybe I do get it, but it's just baffling to me that there's so many people out there who just are against vaccines. I'm okay with people being worried about it. I'm okay with that. Well, I am not okay that they just simply reject it because, you know, they're a Trump supporter. That just doesn't make sense to me. Are you with me? Yeah, that too. Yeah. So I didn't really get on Facebook all that much, but now I don't get on almost at all. I have a page for this course, and I just don't want to get on it anymore because there's just so many people telling lies nowadays. And in the United States, if you tell a lie enough times, it becomes people's realities, right? Even though it's not true. And even when you go on and try to help them understand, and they just simply think you're a know-it-all, and they will, I don't know, I guess you guys say trolling. I guess they troll you and basically say you don't have the background to be able to talk about this stuff. I'm like, oh yeah, I do. and and then they kind of call you bad names. Are you with me? So we need to be able to understand this. And as healthcare workers, you guys are going to be on the front lines when we have another pandemic, right? In my lifetime, I've been around for 50 years. There have been 5 pandemics, 5, right? And nobody knows about them except people like me who are nerdy enough to want to know about them, right? I mean, I love Nelda to death, but Nelda doesn't watch the news. Are you with me? So how's she going to know when there's problems? Nelda, I'm playing with you more than anything. All right. All right. Okay. So, but I get it. I get it. I get it. The the news is sad. There's almost never any good news there, right? This hurricane killed this many people, right? There was an earthquake in Haiti. Uh, there's a nuclear fallout here. You know, there's never any good news, right? We never really talk about the good things that are happening because there are a lot of good things, but we tend to dwell on the tragedy that is the human experience, right? And therefore, we just simply um, don't know any better. So I'm going to talk about COVID-19, and then we're going to stop for today. Um, and we will talk about HIV/AIDS next time, and then we'll start Unit 4. Okay. So let me go through this. So for those of you guys who are tired of hearing about COVID-19, I'm going to give you information you probably haven't heard yet. So it might be to your benefit to just kind of listen to this a little bit, right? Okay. We do have lab today, Jordan, and it's going to start, I think I said at 12, about 12:45 or something like that. Okay. All right. Jordan, um, you know what you're doing, so if you have to go, go, okay? All right. So COVID-19 was a name given to the virus by health professionals so that we could talk about it and everybody could understand it, but it's not its real name. The real name for COVID-19 is severe acute respiratory syndrome, and then coronavirus number 2. So it's SARS-CoV-2. That's its real name. You need to know that for the exam. Okay. COVID-19 is its nickname, but that's what people want when they are talking about it, because you know SARS-CoV-2 is just too hard to understand for most people. It's called a coronavirus because if you look at an electron micrograph, the electron micrograph shows this crown around it, right? And so therefore corona means crown. And so that's how it got its name, right? It has a lot of really important parts to it, right? The most important part is the spike protein, right? The spike protein is what attaches to our ACE inhibitors on our cells, and it basically tricks the cell to saying, let me in. Abracadabra, let me in, right? And the cell opens up when that spike protein attaches to the ACE inhibitor on our surfaces of our cells, and all of our cells have got it, right? And so therefore it gets in. It has a lot of other parts, right? It's a RNA virus, it's enveloped, right? Well, those are the important parts that I want you to know, right? So the spike protein, it's an RNA virus that's enveloped. Those are what I want you to know, okay? Lots of other parts, but not important for this course, okay? Some of the symptomology we see is a dry cough, fever, tiredness, difficulty in breathing, loss of taste and smell. And that loss of taste and smell has now become the more significant symptoms of having had COVID-19. And in some cases, the loss of taste and smell can linger for months, right? There's a person who's had it who got through COVID-19, and that was about 4 months ago, and still doesn't have his taste and smell back, right? Most people, about 80% of them, will recover from the infection of SARS-CoV-2, COVID-19, without any special treatment, right? But the people who are most susceptible to contracting and dying from it are older people, people with asthma, diabetic patients, people with heart disease, cerebral vascular disease, the morbidly obese, smokers, anybody who's got any kind of COPD, right? Whether it's emphysema or whatever, right? And then pregnant mothers, right? Those are the people who are most likely to be affected by it, right? COVID-19 has changed our lives forever, right? There's telemedicine now, it's very popular. The way we handle friends and family nowadays, there's no more handshaking, not that I know of, right? It's fist pumping, it's a kind of waving, but not, right? The way we meet with people, the way we teach. And even though I can't wait to go back into the classroom, a lot of my colleagues don't want to go back to the classroom. That's right, hugging. I'm a big hugger because I'm Hispanic, right? The way we dine, right? So most of us pick up food now. My wife and I don't go to restaurants anymore. We pick up food, we eat at home. The way we shop, Amazon's a big thing, right? Our personal interactions, the way we vacay, right? Curbside services. It's not going to be free anymore. It's going to be considered a privilege. You're going to have to pay for it, but you're going to It's still the way we vote. Well, maybe not. I don't know. I thought we were all going to do mail-in ballots, but it doesn't look like that's going to happen. Uh-huh. So good for you, James. I am ready to go too, but my wife doesn't want to let me. So yeah, gardening is a big thing. Baking is a big thing. Family time is back, and so uh, during COVID-19, we saw families cycling together, picnicking together, walking together, and hopefully that'll still be around because it's still happening in my neighborhood, right? So it's an amazing thing that it's changed the way we do things, right? But yeah, there's lots of ways to. Well, okay. Yeah, you're right. It has slacked off a little bit. I mean, there are big parties now. I play tennis every Wednesday night now with a bunch of friends, but we've all been vaccinated. And as a matter of fact, we met as a group and we wanted to know what the rules were going to be, and we came up with our own rules. And one of the rules was that everybody who wanted to be in our league had to be vaccinated, and one guy didn't want to be vaccinated, right? And we told him, well, then if you aren't going to get vaccinated, you cannot play with us anymore. And he got up and left, and we haven't seen him since then. But you know, we have a lot of older people in our group, and yeah, that's true. That it doesn't. That's right, Nelda. You won't. That doesn't mean you won't get it, but we know that if you get it, that the time of having to deal with the viral infection is truncated. It's shorter, and it's less severe, right? Yeah. So that's an important thing to think about. I can remember people, not very smart people, telling me things like, well, it's just like the flu. I'm like, no, it's not like the flu. It's very different, right? RO means reproductive number. It's how easily it spreads from one individual to another. So for flu, if you have it, you can spread it to about another person. If you have COVID-19, you can spread it to possibly 2 people, right? So it's much more communicable. Incubation time for flu, 1 to 4 days. For COVID-19, 1 to 14 days, right? Different, right? Hospitalization rate for flu, 2%. For COVID-19, 19%. How is that the same, right? Case fatality rate for flu, 0.1% or less. In the United States, about, let's say about 2.5% case fatality rate. People die, right? Because they come down with, so everybody who has COVID-19, 2% of them are going to die, right? Now that's different in different parts of the world, right? In Italy, it's up to 11%, right? There's a lot of older people there in Italy. And in places like Missouri and Arkansas, they're having a big case fatality rate because there's a lot of non-vaccinated people in there, right? Yes, Justin. Yes, sir, Justin.
For the hospitalization rate, is that how often you see it show up in the hospital, or is it of the cases that are in the hospital, the ones that get admitted?
It's the latter, right? So it's of all the people who come down with COVID-19 in that area, that's how many people get admitted to the hospital, right? Yeah. Okay. So here in Austin, our cases are really low right now. I don't know what they are. I haven't been keeping up with it, just because it got below a certain threshold, and I'm like, phew, it's time to go play tennis again, right? I'm not going to be afraid anymore. I still wear my mask in public, not on the tennis court, but when I go to HEB, I still wear my mask. And when I wear my mask, I consider that to be a respectful thing to other people, right? So that doesn't mean that I'm mad at people who don't wear masks. That's not what that means. It just means right now that if I have COVID-19 for some reason, I don't want to be the cause of somebody else dying. So I wear a mask until I feel comfortable with the rates that are in Austin, Texas, and then I probably won't wear a mask anymore. So when I go back to school this fall, if the numbers have fallen to a significant number that I'm no longer worried about it, I'm not going to wear a mask in my classes, right? So, but I have my vaccine passport. I've got a picture of it, and so if anybody ever wants to see it, I can pull it up on my phone and show it to them real quick, right? That's how serious I am about trying to solve this problem. If to me, if you are unvaccinated, you are the problem. right? You are not helping solve the problem. Okay. We know that the coronavirus can spread. We used to say it's 6 feet, but we now know it's 12, right? So it can go up to 12 feet. And so that is a problem. We know that it is attracted to the ACE inhibitor, the spike protein attaches to the ACE inhibitors. I'm sorry, the ACE receptor sites, and it tricks the cell to let it in, right? From our understanding so far, there was a mixing of proteins between a bat and a pangolin. This is a pangolin, it's pretty cute, right? And humans. And so that's what made it the novel coronavirus, right? The little N in front of it means novel, means it's never been seen before, right? And so these are the 3 different hypotheses that we have. And so people are studying that. There is another hypothesis that that it got out of a lab because people were doing gain of function research. And what that means is they were making the virus more dangerous so that we could understand it better. And so I am not against looking at that hypothesis. I think people should be studying it, because if it truly happened that way, then there's some people that need to be held accountable, right? What I will tell you is that there have been over 2,000 different variants. And right now we're looking at the Delta variant out of India as being the most severe, right? And it's right in this area right here. So the Delta variant is really, really problematic, right? It's dangerous, right? And so you can see how these different organisms, these different viruses, there's the one that is the problem, right? Increased severe illness and death, increased transmission. But look at that. If you are vaccinated, there's minimal impact so far, right? That doesn't mean it's not going to change, right? We believe that it started in the Wuhan district of China and it spread from there. Is that for sure? 80%. Don't know for sure, but 80% of us think that. And I'm open to studying it more to see where it truly came from. The problem is certain countries won't give us information, so we won't know, right? So the case fatality rate by numbers, right? And this is out of China, but it could be extrapolated to just about every country, right? So look here, if you're 19, 20, or in your 30s, no need to really worry. If you're in your 40s, not really need to worry. But once you hit 50, the case fatality rate goes up significantly. So what that means is, for those of you who are very family oriented, like my family, and you have an individual like my cousin who won't get vaccinated because he listens to Tucker Carlson all the time, then we just basically told him that he can never come to family functions ever. He said, I'll wear a mask. I said, I don't care if you wear a respirator. You are not welcomed at any of our family functions. And so he hasn't come. I would punch him in the mouth and get rid of him, because there are too many of my family members that are elderly and they are at risk. Okay. So you can see the problems are in those areas that we have, and this is an older map, but where we have more cases are going to be the areas of the country that are not vaccinated, or that individuals have to do kind of service type of you have to do service type of jobs, bus drivers, that kind of stuff, right? And so you can see where the problems are in the United States, right? Other research has basically shown that if you are type A blood, you are more likely to develop serious infection than any other type. Statistically, that is significant, right? That is basically saying if you have type A blood, you should be really watching out, because that's a problem. If you have type O blood, like me, it's protective, right? And so people with type O blood are less likely to get infection. Abdullah, you might be interested in this because you're interested in those kind of things, but here's the reference that I took it from. You can read that paper. It's pretty cool, right? The things that have worked so far are remdesivir, right? It was a huge population study, yeah. The convalescent plasma, right, worked. Two of my family members called me and said, what do you know about this convalescent plasma? I said, it works. She said, okay. So they're a married couple. They got COVID-19. They went and they donated their blood for convalescent plasma, and I was really proud of them for doing that. Dexamethasone is a steroid, but what it does is it decreases the inflammation that you see associated with COVID-19, and so therefore it doesn't allow for the disease to be as bad. Yes, sir, James.
To donate convalescent plasma, you have to have actually contracted COVID, or could you have been vaccinated and then donate?
No, you have to have had as I understand it, James, you have had to have had COVID-19.
Okay, that's what I thought. Thank
you. Yeah. Okay. All right. So, so this, I'm not going to even talk about that because we just need to move on. But vaccines are important, right? The 3 vaccines that are in the United States, 2 of them are messenger RNA vaccines, right? That's the Pfizer BioNTech one and the Moderna one, and the other one, of course, is the J&J one, which is an adenovirus delivery package, right? They all seem to work really well. They all have problems, but the problems are very rare. And so, if you feel like, to me, the safest one appears to be the Pfizer BioNTech one. I am not a physician. I'm not giving you medical advice. So, but that's the one I took because I did my research. And when I figured out that it looked like Pfizer BioNTech was the safer one, that's the one that my wife and I took. And we're pretty happy with what's happened, right? Okay. So, if we look at just the hospitalizations and severe cases from January this year to just May 19th, right? There's been a complete change in the country, right? We are almost through it. And so I'm really happy for that. Okay. So I think that's it for today. Oh, I do want to say that there were some bombs. That whole chloroquine, hydroxychloroquine, azithromycin thing that was touted by certain people was a bomb. There were 3 clinical trials that tried it, and they had to stop all of them early because people were dying from it. And so don't let anybody tell you anything different. But I do want to tell you that the largest spreader group are the 20 to 35-year-olds, mostly because I think they're just tired of being locked up and they want to go and have fun. And I don't blame them. I mean, I always wanted to have fun when I was 25 to 35, right? So they want to have parties and be with their friends and all kinds of things. So, but they are the largest spreader group. Okay. Ladies and gentlemen, we will finish virology by talking about HIV next time. We're going to move directly into the lab. And so go ahead and get out of this and then come back to the lab and we'll do lab